Welcome to The Whole Pineapple. I'm Ruby Boris.
And I'm Ann Judge. The Whole Pineapple is a podcast about wellness focused around fertility. We'll talk with experts on a wide range of topics, including nutrition, stress relief, sleep, mental health, etc.
And we'll dig into the issues that can cause fertility challenges, like PCOS, or early menopause, or cancer treatment.
So if you're curious about your fertility, whether you're ready to start a family,
or you're preserving your options for the future, let's dig in. Hello, everyone. We have reached our next stop on our tour of the fertility clinic, and I'm really excited for this one. I think this is the part of the clinic that most people are probably the most curious about.
Yeah, I think it's kind of like that black box of mystery where it's like, somehow my eggs go in and then an embryo comes out. So
Exactly. And it just happens to be a top contender for one of the most fun departments in our clinic anyway, or at least it seems to be.
They definitely look like they're having the most fun. I'm always so jealous when we used to have our all staffs for the different departments would put on skits. Somehow the lab always had time to have this like elaborate professional costume skit, and the rest of us were like, oh, we were supposed to do a skit. Nursing was too busy.
Yeah.
So
I don't know how it happens everywhere else, but at least our embryologists are so smart and so cool and so nice and just all around lovely humans. And I wish I could hang out there. I never get to see them because I'm in Tacoma and they're all in our Seattle office. But in other words, the best kind of energy is hanging around your eggs and sperm and embryos up there. So we're talking with one of our embryologists today. So Julia Aliabadi. And she graduated from the University of Washington in 1992 with a zoology degree, and then got a job in an andrology lab at Virginia Mason back when they had a fertility program in 1992. She joined SRM in 2005 when the fertility center at VM closed. And she's been with us ever since. And we're so excited to have her on today. She's wonderful and knowledgeable and so cool.
Oh, wow. Such an introduction. Well, I'll come here all the time for those good words.
Yes, thank you so
it's
much
always
for
a
being
little
here.
funny to me because when I talk to people who are embryologists, like there's such a high percentage that started out in zoology. And so I kind of feel like, is everybody that majors in zoology either planning to work in a zoo or planning to be an embryologist? Because I never met anyone in other fields. So.
Do you guys spend a lot of time on sperm in zoology courses or something?
No,
like that?
not one bit. You memorize all of these, different kingdoms and phylums and, all this for animals and insects and stuff. So I never thought that I would be in this field at all. I graduated and I thought I'll have to go back to school. So I need a job in order to, work and make money so that I can go back to school and get my master's degree or a teaching certificate or something. And then this job at Virginia Mason, I basically just applied to be sort of an office person. And they said, well, you've got lab experience because I had worked in a lab during college. And if you have lab experience, maybe you'd like to do this.
Wow.
And I was like, what? Okay.
So what were you thinking you were going to do when you were majoring in zoology? Or did you not really know?
I did not know. When I went to school, I wanted to be a physical therapist.
Okay.
And I knew that when I graduated from high school, I was going to school to be a physical therapist. And then once I got into some of the classes, and some of those huge classes and courses are just weeder courses at the U. Like if you don't get a 4.0 in it, you might as well not even try, because all of those people are going on to med school or physical therapy school or becoming nurses and, stuff like that. And I was okay at it, but I wasn't really, really good at it. So my testing ability was not so good. And then I started volunteering at different physical therapy places, like at Children's. I volunteered at their physical therapy department.
Okay.
And it was hard.
Yeah.
It was hard. Physical therapy school at the time was more competitive than med school. So I just knew that I wasn't going to be able to get in. And so I thought I'll just get out of college with a degree, in the 4 years that it took me, and I graduated.
And when you had your lab experience in college, did you like being in the lab? Like, were you kind of like, this is cool?
Did.
Or, okay.
I did. It was a weird job. I had to take care of a mouse colony, and the mouse colony was in a building that's in the quad at the U, which most of the buildings in the quad are sort of like, they're arts more and stuff like that. And we had this mouse colony that I had to take care of. And we were doing studies on cholesterol in mice. So I had to, take care of the mice and then do some testing on them. I never had to actually kill them.
That was my next question.
Yeah.
Some of my roommates from
Oh.
college went into med school. One of them was like, I can't even think about the mice that I have because she did neurology. She's like, I've looked at so many little mouse brains and I had to be the one to end them. And it's like,
Oh, no.
I know. That's like the worst thing. And so when I was in school, I never had to do that. I mean, they just kind of ran their course and then sort of like disappeared.
They went to a little farm at the side of the U where all the mice live happy lives.
Exactly.
An endless tube and a little runner thingy.
Exactly. They're making more mice in the quad or whatever. But then I thought that I would go back to school and maybe I would become a teacher. My mom was a teacher. And so I was like, I think I can probably do that. But then the job in andrology came along and I had like one of the best bosses ever. And she just taught me everything that she knew. And then I started killing mice and hamsters.
Oh, no.
I mean, that's not the fun part of the job. And now we use everything. We get things like, frozen mouse, and we just use them for testing and making sure all of our equipment is running.
Yeah, I think people are sometimes surprised. Like you read about old things like the hamster penetration. Like you forget that there's overlap with animals and what we do in humans. So I didn't realize that we're still using rodent stuff even now to test things. Is that pretty standard?
It is pretty standard to use mouse embryos for testing plastics and things like that. And also as a sort of a control for our frozen tanks. If we thawed mouse embryos and they, somehow didn't survive, we'd know maybe something had happened in the tank or something like that. So it's usually mostly as a control, but we don't use them on a daily basis.
So that zoology degree really does make a lot more sense than people might think. You're still dealing with other species.
Yes.
Okay.
Exactly.
And for our listeners, just so you know, like the lab is extremely regimented and protocoled, and there's lots of controls and backups and all sorts of things. And I'm sure we'll talk about that a little bit more.
Yes, there is no chance they're going to thaw a mouse embryo when they're supposed to be thawing your embryo. I feel like I have to put that out there since now it's one more new thing for people to worry about. So.
There's no chance. No chance of that. None.
So when you got to Virginia Mason, you were doing andrology, which is more on the sperm side of things, correct?
Yep. Semen analyses, IUI processings, as well as, testing like the sperm penetration assay is what we did there. I think a lot of places don't use that much anymore. I mean, ICSI kind of takes care of that in the in vitro situation.
And so for people that don't know, can you explain kind of what they used to do in terms of the penetration and then what ICSI does now?
We would use the patient's sample and process it, and we would put it in with a hamster egg that had no zona. So if it had a zona, then it would stop the sperm from penetrating. So basically, we would count the penetrations of this zonaless hamster egg, and we would extrapolate that to say, this specimen most likely could penetrate a human oocyte, and then give them sort of a knowing that if they just did standard insemination, that they could get fertilization.
Which I think probably blows people's minds. It's like, you think of that we still have a ways to go in fertility, but at least we're not just putting sperm in with hamster eggs and seeing what happens to get an idea of the potential for sperm. So.
And the worst case scenario is you do standard insemination and you have failed fertilization. And this was a way to say that we should probably use ICSI and just make one sperm go into the egg to attempt for fertilization.
So were you working at the time that ICSI was now suddenly becoming the new standard? Did you get to see that? Must have been so exciting to see that transition.
It was exciting and it was frustrating too, because it takes a while to learn it.
Mm-hmm. So maybe explain what ICSI is for people that don't know.
So ICSI is intracytoplasmic sperm injection. So you will take one sperm and you're using a holding needle to suck your egg onto the holding needle, and the egg has to be a mature egg, which I know we're going to talk about. It has to be an M2, a metaphase 2 egg. So, and then you catch a sperm with your ICSI needle, and then you insert that sperm into the egg, and then you wait until the next day to assess for fertilization.
I remember being surprised the first time I got to like come in the lab and see with how kind of violet it looked though to catch that sperm and like kind of get it ready for ICSI. I was like, wow, it's like this hunt for you to figure out which sperm you're going to take and then to like get it in that perfect position.
Yeah, come back here.
You understand then why it's not something that everybody can do, why like it takes a lot of training to get embryologists that are going to be able to do this in a way where you're going to hopefully be improving your chances and not destroying the egg by doing ICSI incorrectly.
Right. Because that does happen, all eggs are not created equal. Not all eggs will fertilize. And we sort of use this analogy, you can lead a horse to water, but you can't make it drink, right? You can lead a sperm into the egg, but sometimes it just won't fertilize for some reason that we don't know yet. But there was a few iterations before people started just pursuing ICSI. They used to do something called SPICSI, I think it was, where they would put several sperm into the paravitelline space. So they'd go through the zona, but not through the plasma membrane of the egg. And they'd let these sperm swim around in the paravitelline space, and then hopefully one of them would fertilize.
So it's a layer in between two different layers of the egg. And the zona, what we mentioned before, is kind of, for lack of a better term, the outer shell-ish. We don't have shells like chicken eggs, but shell-ish.
I always think of it kind of like a gobstopper, how it's like the extra layers as you get to the middle. And so you got like different rings of different flavors around the outside. And so...
I will never look at gobstoppers the same way ever again.
And so in what we might think of as like standard insemination, or when a sperm is finding an egg on its own, whether that's inside the body or in that microdrop, usually once that one sperm has made it all the way inside, that's what kind of hardens out the rest of that. So that's what's going to keep other sperm from penetrating.
Exactly.
And so with ICSI, you're basically just doing that all by yourself. You're putting the sperm all the way in.
There was several attempts at trying ICSI because you didn't know, should you put the tail first into the egg? How aggressive do you need to immobilize the sperm? Because you have to inactivate the sperm in order for the sperm to undergo its sort of capacitation steps. Because the sperm, when it's traveling up through the vagina and into the uterus, it goes through this capacitation process where it's getting ready to fertilize an egg. And when we immobilize it during ICSI, we really have to make sure that those steps are happening, because if they're not, then the egg won't fertilize.
Yeah, listeners, if you've never seen it, you should go online now and search for video of ICSI, because it's like you're lassoing a sperm and then just whacking it against the fence almost to like make that. I just remember being like very surprised by how active a process it was to get that sperm ready, and watching the sperm kind of swim away in the beginning. Like it is, life is a struggle. You see all these tiny stages that have to go right, even when we have egg and sperm in the lab and can see what we're doing, as opposed to when it's all happening mysteriously inside a fallopian tube.
Right.
So you got to see that whole transition to ICSI, and that was huge in the field, because before that time, we were having good success for people who had blocked tubes with IVF, but you still had to have enough sperm to fertilize an egg in a dish when you just put them all in together. And so for those with really low sperm counts, you had very poor success rates even with IVF. And ICSI really opened up this whole new area for those that had really low sperm counts, which has been amazing. Although it's always interesting when you go to conferences and you hear urologists talk, how they're kind of like, ICSI really set our field back by decades, because once we could do ICSI, we stopped trying to figure out if there was any other way to treat really low sperm counts or other quality factors that we might not have been aware of. So yeah, it gave us this like new tool to have good success rates, even with severely low sperm counts, but it also kind of stopped us from hunting for other ways to maybe improve whatever the root cause was.
That's very true. We just bypass it. Go straight to the testes, get some sperm, and have our way with it.
Yeah, and we only need however many eggs we have, we just need that many good-looking sperm to go into those eggs. So it does make a huge difference when you have, only dozens of sperm compared to millions of sperm.
So what kind of training did you go through to learn how to do all of this? I know the field is different now than it was back then. Was it all just kind of on the job? Someone would mentor you and show you how to do it?
On the job. That's the way it was. At the time, my lab director, she had gone, Jacques Cohen was kind of the founder of ICSI. And so he would put on these symposiums and people would go and learn how to do it. And then they'd come back to their own lab and then try to redo it. And we had to make our own injection and holding pipettes.
Wow.
We had machines that would pull them apart. We had things that would grind them down. And luckily, the amount of things that we were doing was not as much as today. Like, we maybe had 2 a week or, that kind of thing. Like,
I'm just trying to imagine doing all of this stuff.
Yeah, but we had to make them and then keep them and sterilize them and then use them. So that was really, some days you'd just be sitting at this forge pulling pipettes and hopefully they would be work.
I think for those of us that have mostly worked in the field, like nowadays, obviously we're still trying to get improved success rates, but you kind of forget what it was like in the early days. At this conference that I was at earlier this year, our own Dr. Lin was presenting and he showed this video of an early like 60 Minutes report on quote unquote test tube babies, where they were going around and interviewing these doctors and talking about, and you claim you could have up to a 30% success, but it looks like it's really only 5%. I mean, like all this, and people were like, yeah, but still 10% success. Isn't that amazing? And it's true, if you have people that don't have really a chance otherwise, that does seem like a good odds. So it's really kind of mind-boggling to think how much the field has changed even in just the time that you've been working in it.
Exactly. It really has. You're able to help more people that never would have had a chance to have success. And I always think about it too, like if it increases 10%, well, that's 10% more, but you're really only going to be 100% pregnant. You're like not going to be 10% pregnant.
Yeah, I know. We always talk it a little bit like it's like the scene in Dumb and Dumber where it's like you're telling all the stats and they're so low and the Jim Carrey's like, so you're saying there's a chance. Exactly. Like, okay, but there's a chance this could work for me. That's what I'm going to focus on.
It really feels like that some days, doesn't
it? Yeah.
So that's, so many things in the field. And that's when and was it kind of the same on the job training in terms of all the other stuff that you started doing too? Like once you're working with embryos and then going to day 5 culture and all this stuff, which is sort of someone in the field advances, they publish, they say, hey, this is a good thing. And then you just kind of learn and grow.
And then we try it. And a lot of times it's like side by side sort of doing things. When we started doing embryo freezing, when freezing started really sort of becoming viable, we'd freeze on day 3, and then sometimes you'd freeze some on day 3, culture some to day 5. If you felt like, they looked good enough, and then hopefully you'd have something to freeze on day 5. The freezer that we had back in the day took like 3 hours to complete. So you'd have to do all your freezing at one time because you couldn't like run it more than one time during the day. I mean, it sounds crazy, but the volume was not really high there either. But things started to change with media, and we started, they started adding more sort of nutrients to media so you could culture to day 5 and get good results.
I think part of the reason that it seems so mysterious for patients nowadays is that we've learned that we shouldn't bother the embryos that much. And so when I started, we'd give you a day 1, a day 2, a day 3, a day 5. And so people would get upset on day 4. Like, where's my embryo update? What are my embryos doing? And so to now go to basically like we see if they fertilized and then we look again 5 days later, because every single time you're taking a dish to look at it, you're disrupting. But in the olden days, I think people did get a lot more information. They got these day-to-day updates on their embryos, and I could see how that was in the day really reassuring, but we certainly continue to learn more as we go along. So
we sure do.
Interesting, and showing the window here, because Anne, like you did, it sounds like you guys did even more from when I started. Then we did the fertilization report the day after the egg retrieval. You got a day 2 update, and then you didn't get anything. Or you got a call with scheduling. Yeah,
when I started, we did a lot of day 3 transfers still. So we'd be called, we would literally, I just remember I'd be working on day 4 and someone would be upset. They're like, what's my embryo update? And I'd have to explain, we actually don't look at them on day 4. And people would be like, oh, but what's going on? How do I
know.
know if it's still dividing?
They're just hitching a ride out of the lab. Don't worry.
I got a call when I was a patient from Embryology. Embryology used to call and give the updates. So.
I was just going to say, we used to do all the calling. Here's your fertilization report. Calling on day 2, we should do a day 3 transfer. Or no, we should do a day 5 transfer. We used to do all of that calling. And like I said, the volume was not there and there weren't as many of us there, but we used to do all of the patient contact, which I thought was awesome.
Does part of you miss that a little bit?
Yeah.
I can see that, especially since you started off working with people.
Right?
So
I do miss it. We don't get a whole lot of patient contact, but nursing has to answer so many questions about things that they perhaps don't know all the information that the patient wants, kind of a thing. But yeah, things have really changed.
And in some cases, there's just information that we all want, but we're not going to be able to have. And some of that happens between day 3 and day 5.
Yeah.
Well, once we're tossed around day 3, day 5, why don't you go back and walk us through a little bit, like, what is basic embryo development? Because I think that's what people are really wondering. Like, I'm not getting any updates. What's actually happening with my embryo?
Right. And it is, just kind of basically, we have that chart on the wall in the embryo transfer rooms of the pictures of each stage of, on this day, you should expect this. And on that day, you should expect that. So on, day 0 is considered the egg retrieval. Day 1 is considered the first day of an embryo where the embryo shows 2 pronuclei.
Let's go back to day 0 because you mentioned before, how do you know if an egg is mature or not?
So if you're doing ICSI on day 0, we will, after the eggs come out of the follicles and we count and see how many there are, we do this process called stripping.
Which is of the egg, not the lab. The lab is not doing any stripping.
I was just going to say.
Unless it's really warm in there. And we have these tools called strippers, and we use the stripper tools to do this.
So every now and then someone's like, oh my God, are we out of strippers? Did someone order more strippers
Erotic
for the lab?
answers.
So, okay, awesome.
I don't know who thought of that, but actually, like in ideas, that is an event that is called stripping.
So someone is the assigned stripper for the day for the lab?
Well, that's actually my favorite job in the lab is stripping. I don't know what that says about me, but to me, it's like unwrapping a present.
Yeah.
So we use this manufactured media called hyaluronidase, or sometimes it's called in vitro or strip something or other. Like they have all these creative names for it, but we purchase it and then the eggs are stripped of those cumulus cells, which are how we can see that there's an egg in the media that comes out of the follicle. So we have our eggs together and then we take all those cells off there and then we group them into whether they have a polar body, which means that they're in metaphase 2 of their development, of the egg's development, whether they're an M1, which means they do not have a polar body, or whether they're a germinal vesicle, which means that most likely they are not mature, they're very immature, and we cannot fertilize that type of egg.
And this is often the part that makes people's heads explode because you're trying to remember your high school biology, mitosis versus meiosis, and you're like, wait, what is a polar body?
Right.
So
metaphase 2 of meiosis, right, is a mature egg, and that is the egg that will fertilize.
And the reason that eggs have to do that to be able to fertilize is what? Can you explain kind of what a polar body is and why they...
Yeah. So the egg is the biggest cell in the body. And so the egg actually has 2 copies of its DNA in there. So it kicks out 1 copy to be the polar body.
Which is kind of like the trash can of the egg cell.
Yeah. It's like you go here, DNA, and then when the sperm is added, it adds its DNA and it forms 2 pronuclei and actually kicks out another polar body. So a fertilized egg has 2 pronuclei and 2 polar bodies.
And that means just
that
crumpling it up and throwing it away.
Yeah. And that's when you know that egg obviously is an embryo.
And that's why we talk so much with our patients about the fact that we can't test an egg for anything, because an egg, you're only going to get half from that egg, and then you have to have the half from the sperm. And it's how those 2 combine together that is going to then tell us if an embryo is either expressing a disease or if it has the right copy of each chromosome.
Exactly.
An egg is just 1 cell, so you test an egg, that's it. That's all you got. It's gone.
Yep. Yeah, you can't do anything with that, right? We used to do like polar body testing, but that really didn't make any sense either because, you don't really want to take that much away from an embryo.
Don't want to test the throwaway DNA.
Yeah, you can't really tell for sure.
Mm-hmm.
So then you culture that embryo and you watch it divide. It's the egg that's doing all the driving of that genesis of dividing, and it becomes a 2-cell and then it becomes a 4-cell. Sometimes people would freak out, like, why is my embryo a 5-cell? And it's like, well, one blastomere divided, but the other one didn't. So that's how you can have even numbers of cells in your embryo as it's progressing. And we mostly count cells when we're doing day 3 transfers because we kind of grade the embryo as how many cells it should have on day 3. It should have anywhere between 6 and like 10 cells on day 3.
And what are we at by the time we get to like a blastocyst?
Oh my gosh, hundreds and hundreds and hundreds. Yeah.
So
Which is why
crazy.
then we can take those cells from the trophectoderm and test them because there's so many cells that are involved there that are representative of the entire embryo.
And that sense of scale is, I think, what's so hard to wrap your head around, to realize just in those 2 days of development, you can go from being 8 cells to now hundreds. And it's an incredible rate. And that also make more sense why not every embryo does that embryos are going to stall out at different phases.
Yeah. It's really that inverse pyramid, even starting from stimulation and counting follicles on your ovary. Like you might have this range of follicles, but you're only going to get this range of eggs from that. You're only going to get so many mature eggs, so many embryos, and then usually only about 50% of all the embryos that you have will look really good on any given day. So it's very much a cycle of attrition, and that not every egg and embryo is going to be pretty viable.
Yeah. Every step of the way, there's usually some sort of drop off of things. And that's expected and actually normal in most cases.
But it's definitely something that we have to keep reinforcing it again and again, because I think people are often disappointed by how inefficient reproduction is, whether it's happening in your body or happening in the lab. Like it is not as easy as people think.
It's like so crazy to think that anybody actually ever has a baby.
Right. Yeah.
Yeah, and we can physically see it in the lab. So then it really highlights like how delicate and intricate and kind of complex this is.
The other part that was really interesting to me, we just had our lunch and learn on embryo and the new grading system this week when she was talking about how, remember, this process is still happening. So if it's a day and someone's transferring, like the embryo they thought looked the best in the morning may actually not be the better embryo by 3 hours later, that these embryos are still continuing that process. So you do realize how much depends on just when we happen to be looking.
Exactly. It's a snapshot in time. And we have to make the best judgment of grading at that time.
And I think that's another thing to go back to the beginning, that we do give things a little bit more of a chance. When you look and you kind of see the mature eggs, they look again too, just to make sure there's not like a late blooming egg that then kicks out the polar body later.
For sure.
So we do try and give the opportunity to make sure that we see that range of variability where there could still potentially be an egg that can be fertilized, or there could still be an embryo that makes it to a stage to freeze. So
yes, definitely. When we go to do the ICSI on we always put the immature, the M1s into our ICSI dish just in case they have matured, and then we would inject them if they have matured.
And this is also part of the reason why when people are wondering, like, why is my trigger shot such a specific time? It's because we've got to make sure that we have the eggs and the sperm at the time that our team is here. So we have this like whole long day to work with. And that's also the reason that we can't do 40 egg retrievals in one day. Like, that's the reason that clinics have to really keep close track of that lab calendar to make sure that you have the physical space, the dish, and the person available to really work with those eggs and embryos in the appropriate time frame.
Exactly. I know it would be terrible to have 40 egg retrievals in one day. We would not be able, that poor OR would be just
That'd be a lot of
oh
stripping.
my God. Yes, it's exactly.
That would be a lot of stripping.
I know. So
we're talking about labs, like how can patients know if the lab for their clinic is kind of up to date, if they're going to what's a quote unquote good lab? Like, how do you suggest people try and gather information?
Well, the importance is, being members of SART, being CAP inspected, knowing that it's like not in the basement of someone's house or,
Even though that's how things started out in like the rabbit
right.
hutch of the research labs.
Exactly.
Yeah. Okay.
Right. We self-inspect 1 year and then we have a full like physical inspection every 2 years to maintain our accreditation. It's important to have a lab director that is certified and knows, what they're doing, that you can trust. These days, a lot of, well, I mean, just like with us, we used to be part of IntegraMed, but now we're part of Pinnacle. That having a sort of governing body is helpful too, because, they don't want to purchase a center that is going to cause them more malpractice than, not. And just looking up at the SART data from the CDC and seeing, making sure everybody's reporting everything that they're doing.
I don't think we've talked about SART too much, Anne, actually, which is kind of surprising to me now, but it's a pretty complex website. It's not super accessible.
Well, they're trying to make it better. That was Dr. Lynn's presentation, actually, was what could SART do for you? And they're getting ready to roll out a simplified form that will kind of be a little bit more patient-friendly. Because the problem is, all clinics, they want to make themselves look as good as possible.
Yes,
so there's
exactly.
sort of an incentive to sometimes present your data in whatever is going to be the most glowing way. And so SART is really there to kind of even that playing field and collect the information, and they make you report when you start your cycle, so that you can't only report the successful cycles and pretend like you're doing less. So they're really there. And that was why he showed this original 60 Minutes video, to show that, hey, before there was SART, clinics could claim whatever they wanted. They could claim what at the time seemed crazy, a 30% success rate, and no one had any way of checking out if that was true or not. And so that's why by having SART and, going to a clinic that does report their data, that there's at least this level of quality control, that when the lab is claiming a certain success rate, or the doctor is claiming that their lab has this success rate, you can actually go back and you can see, well, is that in my age category? Or are you quoting me the success rate for people who are 30 to 35, but I'm 38? So it really is a way of helping patients understand a little bit more about what are the actual outcomes from that laboratory in that clinic.
SART, it stands for Society for Assisted Reproductive Technology. And if you're curious, it's at sart.org.
Yeah. And they work well with ASRM too, the Association of Reproductive Medicine. You can find links on that site as well too.
I don't think I've ever heard anybody call ASRM that before.
I know. I was like, do we call it ASRM? I've never called it that either. I was in my mind, I was like, must look up ASRM.
Only the cool kids do.
Now you will.
Now I definitely will. I want to be like Julia when I grow up.
You have a ways to go. Yeah. So it's important for patients to know that they have options, that they don't have to just go to whatever pops up on their Google search, and to investigate.
I remember speaking once with Dr. Carnegie, and she read a letter that a patient had written her that was thanking her for her care and talking about how, I trust you with the most important things in my life, like my eggs and my hopes and my heart. And so, I mean, the trust that you're putting in a laboratory is huge.
It's huge. It's huge. And we do take it very seriously. I mean, you kind of, from the first step of like setting up for the next day, being very careful about making sure everything is set up. A misidentification is our nightmare. Everything is like double, triple checked. We have multiple steps involved in making sure that everything is in the proper chain of custody. But, once you get those eggs in the dish, they look like any other egg, right? And so you just have to really be very careful.
How are you physically labeling things? Because you're right, we're talking about tiny, tiny little straws. Like, how do you keep track of like what dish and what pipette and all of that?
They each get a little name sticker.
Yeah, it's a sticker with the last name, the MPI,
which is our medical record number,
and then a color code. Yes, medical record number. Yep. Yeah. So, and those are all the color code matches, and we put we have our incubators, and there's a white index card with the patient's last name, their MPI, and their color code, and that follows those dishes wherever that dish goes, which is not very far in the scheme of things. But yeah, and we have multiple identifiers. Like if I'm going to sit down and do an ICSI, someone is following me and saying, and I say, I'm going to use this sperm because it's labeled with the same things as this dish, and that it all matches, and we sign our initials that we've looked at it and it matches, and then we can proceed. And the lab is a fun place to work because we're all great people, but we pay very close attention, and it's a high stress level as well too. You pretend that the patient is standing there watching you do everything. So that adds an extra level of intensity to it, but we do what we're supposed to do. And even though we are dealing with like the most precious things, it is our job, and we're very professional about what we do, how we proceed.
For us, there's only one day of the year that our clinic is actually physically closed. So the protection of these embryos and sperm and eggs happens all year round. And one of the questions I get from a lot of people, especially more so people that are freezing their eggs, I feel like that's come up a lot is in my egg freezing patients, is where do they get stored and what are the backups in case something happens to where it's stored?
We have so many tanks in the lab. We have 5 really large tanks that have up to 65 canisters in each tank, and there are 12 patients that can fit in each canister. So, 65 times 12. And then with eggs, we freeze usually 2 eggs per container. You can fit 9 straws, so to speak, or devices.
And give a sense of scale. How big is a straw or device? Like when we're talking about what's being frozen.
Yeah, this makes it sound like huge.
Right? A straw would probably be, like a regular straw, and it's probably in diameter a quarter of a regular straw.
So like a Starbucks straw?
Yes. So you can probably fit 4 of our, like we call them cryo tops or whatever we buy them. You can probably fit 4 in one Starbucks straw, like in width and diameter.
I think a lot of people are thinking of it like Jurassic Park, like how Nedry has the shaving cream can,
right?
And that has like 10 dinosaur embryos, and so I don't think people realize how small.
And you have done a few things this episodes that are like, tell me how old you are without telling me how old you are.
That's true. What? People still watch Jurassic Park. It's a classic.
For sure.
But I think honestly, as a kid, that's probably my first exposure to embryos, right? That you had this movie that was like, oh my gosh, we can in a lab create like embryos and transfer them. So dinosaur embryos, how cool. So, but what we're using now is much smaller. But also, that's in the back of my mind when people ask about shipping all the time. It's like, because sometimes people are like, well, what if I freeze my eggs, but then I move? Shouldn't I have my eggs follow me around? It's like, well, there's always that chance of something going wrong in shipment. And, these do need to be stored pretty precisely. And so, anything we can do to minimize potential risk is what we want to do. And they don't need to have everything follow you around until you know if you're actually going to need to use eggs or embryos.
We store them in actual liquid nitrogen. So everything is kept in liquid. So we've got these 4 big, huge tanks, and then we probably have like 30 smaller tanks all around on, they look kind of little like R2-D2. And so I guess tell me how old I am, but don't tell me how old I am. So they're kept there. They are submerged in liquid nitrogen, and we have temperature sensors on the tanks and in the tanks that are monitored 24 hours a day. And if an alarm goes off, we all get a notice that there's an alarm that has gone off. The incubators are also alarmed as well as the front door. Like if someone goes into the embryology lab without access, like if they walk in there, that's happened before with the cleaning people. They just somehow got access to it and they walked in, and so there's the burglar alarm went off, and actually the police were called. But I'm close by. I'm from the Seattle lab on Westlake to Ballard, I'm like 10 to 15 minutes away, and I have come in at 2 a.m. to check out an alarm that's gone off, and it's always been nothing. I have to knock on wood, but they're monitored 24/7. There's a huge phone tree of all the people that work in the lab who are all on the phone tree. So it just goes down the list of this person's first, then that person gets called. If no one answers the phone, then it goes back up again and then goes to like other people.
And I've worked in Seattle during some of our big like snowstorm blizzard days, and like people hike into the clinic to check on the tanks and make sure the generators and make sure that everything.
Yeah, I would say I'm sure you don't get very far on the tree, because I every time we've had some sort of issue, like there's always somebody there almost immediately.
And I think part of the reason that we get asked about this so much is it was pretty well publicized a few years ago when there were 2 major tank failures at different clinics. And so when you read the actual reports of what happened, like you see what they call that Swiss cheese model, where you had to have a lot of things line up, where people had turned off alarms. And I mean, you realize like you have these layers of safety check, and it takes a lot to break through those. So
yeah, it does. And I mean, it's such a shame when it happens. We're very particular about how we maintain everything, and with the daily checks and the QC and monitoring. Yeah, it's very important.
I think to wrap up, what other kind of questions do you get from patients? I know you don't talk to them a lot, but like at the embryo transfer or things like that, or what are the common questions people ask you?
The most common thing is, how do I know that this is mine, right? Like, and then we just have to reassure that we, follow our chain of custody. There's several people that are verifying that it is the right chain of custody. If they knew how awful it would be if something got mixed up, how devastated as an embryologist you would feel. I mean, everybody is human. Mistakes can be made. We know this, and hopefully they're not like too detrimental. But, when I was learning, sometimes accidents happen, like I'm always afraid I'm going to trip or whatever, but no one is more devastated than the person that makes that mistake. And, you just know that you are just always trying your best.
I think, that's why we emphasize our protocols and how diligent we are about them. I've had patients get really upset, like if they show up for their IUI and they don't have their ID, and we're like, I'm sorry, like we can't do an IUI unless I know you. I know I've met you 20 times, but we have this policy for a reason, that unless you have the stuff to really confirm your identity, like we're not going to be dealing with sperm and...
And you should ask me several times, what's your name? What's your name? What's your birthday?
Yeah, I want to be asked, like, what's my birthday? What's like, I want you to be looking at my band, comparing it to, the sperm sample for us when we're doing IUIs, but inside the lab, we have 3 checks for every label, and everything gets a label. So, and even at the time of the embryo transfer, they do, when you sit in on transfer, the patient's there and all set up. And before they do the transfer, they actually show the Petri dish in the camera with the label. And isn't there a color too? Like you color code everything too?
Yep. It's our final ID check. It's the last name, it's the MPI, and it's the color. And I've never had someone say, that's not me, which is a good thing, right? Yeah. And then they get to see their embryo in person, sort of on a screen as it gets loaded into the catheter, which I think makes things a little bit more real for them versus
it's
just
really cool.
the picture of it. But
that's one of my favorite parts, is when the embryo just is kind of like floating up the catheter.
Yeah, it is pretty cool. It's pretty cool to be able to share that with the patients. And so, I think that's one thing that, people want to make sure that it's theirs. Sometimes people say, can I pee the embryo out? And no, you can't. And, it's either going to implant or it's not. And that doesn't mean anything other than it just sort of like fades away. I mean, I don't know what actually happens to an embryo that doesn't implant. Like, does it just disintegrate? I mean, obviously it probably comes out when you have your period because the lining's slough.
I think that's sometimes the part that's so frustrating for people, is they're like, but I want to make sure I'm doing everything right. And it's like, isn't there some position I should lay it or something else I should do to help that embryo implant? And so just trying to explain, like, no, like we're putting it in the right place. But beyond that, it's really up to the embryo.
It is frustrating.
Yeah, I know. I wish it could work all the time. Like that would be so awesome. Or I wish that I could put a camera on that embryo and see exactly, right? See exactly what are you doing in there or what aren't you doing in there? Because it's, it's not like it's this big cavernous space. It's like, we kind of talk about it being like an English muffin, on the inside with all these nooks and crannies on it in the uterus. And
Oh, that's good. I always say a balloon with peanut butter so that people realize it's like, sticky, because I think people think of their uterus as being this like wide open space, and isn't the embryo going to float around? It's like, no. Those walls of the uterus, even though we're measuring it, like it's different layers, but they are all stuck together. And when you talk about tell me how old without saying, I think of Look Who's Talking in that early scene where you have the sperm and trying to find the eggs.
Right.
Yeah, if we had the ability to like watch our little embryo try to orient and implant in the uterus, that would be amazing.
And all the talking that happens. Yeah.
I mean, that movie, like, it has staying power, though. Like, I still remember that scene to this day. So it definitely...
It gives a good sense of scale between eggs and sperm, right?
Totally.
That you have these teeny tiny little sperm and then that giant glowing egg at the end. So.
Right. Isn't that funny? And then like getting even beyond this, I would tell when my kids were getting older and we had to talk about the sex talk and stuff. I was like, nope, you just go to mommy's work and then you have a baby. That's how it's done. Don't ask any more questions.
It's so funny because I always say that I'm like, oh, the sex topic is going to be nothing. I do that all day long. And we're like, listen, sit down. We're going to talk about what I do.
Yeah.
And
and then so much information that they're just like,
Yeah, like, stop it. Yeah.
Any other common patient questions you think we should give an answer to?
People want to know why it doesn't work, and we don't really know that. If we knew that, then we would be able to make it work all the time.
And we're going to talk in our snack about what we call PGTA when we talk about testing the quality of the embryo, because that's, besides ICSI, this has now been the other huge advance in terms of our success rates. But I think it's made it so much harder when it doesn't work, because people are like, well, you're telling me I have a quote unquote normal embryo and that you put it perfectly in my uterus, so how could I possibly not be pregnant?
I know. And that's sometimes more devastating when they do have tested embryos when it doesn't work. But it's becoming more so that it should work than it doesn't work, I mean, obviously not 100%, but closer.
Oh, cool. I can't wait to hear about the embryo biopsy episode. That's like really like we're going to get into like the things that are all over the news and like some of the things that are up and coming and really what happens in that petri dish. So it's exciting.
Yeah, so tune in for our snack episode and we'll go even more in depth on the embryo. So thank you so much, Julia.
Yeah, you're welcome. Happy to be. Hopefully I was informative, and
I learned so much about stripping today. It was great. So thanks for joining us for this week's episode of the Whole Pineapple Podcast. We hope it was helpful.
If you know someone who could benefit from hearing the podcast, we hope you'll share it with them.
And don't forget to subscribe, rate, and review the Whole Pineapple on your favorite podcast app. Every rating and review makes us easier to find.
This podcast is sponsored by Seattle Reproductive Medicine and is produced by Audiotocracy Podcast Production. We'll see you next time. Have a delicious week.